Hello. So today I am going to talk you through the process of growing microorganisms in a lab. Now, I'm going to need you to bear with me a little bit and use your imagination because I don't have a lab available to me. I have my kitchen, and so I am going to be using some alternative equipment to grow my bacteria. Now, the first and a really important thing that you need to do before you start growing bacteria is to make sure that everything you have is completely clean. So the first thing that I need to do is to grab disinfectant and wipe it down. So I'm going to wipe down my surface. I've already washed my hands. If I had gloves, I would also be wearing gloves as an added level of protection to make sure that I'm not going to get any of the bacteria that would exist in my hands into my plate. I'm trying to grow bacteria so that I can study them, and so if I have a mixture of bacteria in there, it's not going to be any use to me. I need the one exact type of bacteria that I'm interested in, otherwise it's no good. So I don't want it to get contaminated from anywhere. So I've cleaned my surface, I've washed my hands, I'm wearing gloves. The Petri dish that I'm going to grow it in will have been sterilised. When I say sterilised, I don't just mean run through the dishwasher or washed up. It needs to be put into a special machine called an autoclave. It's kind of like an oven, except it gets hotter than a normal oven will. And along with the high temperature, it releases steam and increases the pressure. So you expose it to this really, really high temperature and pressure, which will kill any bacteria that are there. So my sterilised Petri dish. I'm also going to light a Bunsen burner and have a Bunsen burner running. There are two reasons why we have a Bunsen burner running. One reason is that we are going to need the heat produced by the flame to help us sterilize more of our equipment as we go through. And the second reason is because the heat produces a convection current, and so it causes the air to heat up and be risen away, and so you're less likely to get bacteria from the atmosphere settling on the table and onto our Petri dish. Inside the Petri dish, I'd have a special type of jelly. Now, it's clear and it isn't anything that you would want to eat, but it is everything a bacteria would need to grow. So it'll have amino acids, it'll have carbohydrates, it'll have some fats in there, it'll have all of the different nutrients that they will need to grow set in a nice gel. So it has a texture of not quite the jelly that you'd want to eat, but a bit more like the cubes of jelly that you then dissolve. So really quite a firm set jelly. In fact, agar, the gel that we make this of, is actually used quite often in vegetarian jellies. So if you have a vegetarian jelly, it's probably agar that is used to make it set. So a very, very similar thing, but with different ingredients in it that is not good for humans, but great for bacteria. And the next thing that you are going to need is a source of bacteria. They tend to grow in little jars, little bottles. If you remember back to when we did our cauliflower cloning experiment, we had those little bottles that we put the cauliflower in. Those quite often will get samples of bacteria in a liquid. And what you need to do is to get a sample of the bacteria onto your agar plate so that they can then grow and you can use that bacteria. And you do that using what you call an inoculating loop. So it's a metal wire with a loop on the end. And again, this is a potential source of contamination. So before you do anything else, you put it in the flame of your Bunsen burner until it glows really, really white hot. And then you take it out and let it cool a little bit. You don't want to superheat the bacteria, otherwise they will die immediately. And you take the lid off your sample of bacteria. When you take the lid off, you don't put the lid on the counter, because then you're contaminating the counter with the bacteria that is in the jar. So you take the lid off, and you keep it in your hand. I am going to be spreading tamarind paste up my gel rather than bacteria. So you keep the lid in the hand with the jar, and all around the neck of the bottle, around here, there is a likelihood that it's been contaminated with bacteria from the environment. And you don't want to get any of that bacteria into your sample, so you run the neck of the bottle through the flame as well. And then you dip your inoculating lube into the bacteria and you spread it on the agar gel. And then you turn it through a quarter, spread it again. You have to be quite gentle at this point. Another quarter turn. That you are just running it lightly over the surface of the agar gel, and then one last quarter turn, rather than really pushing firmly, because you can break it and you don't want to break it. You just want the bacteria sitting on top. And you close the jar. You'll put this into a disinfectant solution, and then you will get a lid and you put it onto your agar plate. So I'm going to rinse this with my hands. And then you'll use sellotape to seal it down at 2 points. Now it's really, really important that you don't seal all the way around the edge. You want oxygen to be able to get in to the agar plate for the bacteria. The reason why you have to be careful to do that is for a safety reason. Bacteria that tend to cause disease in humans like to grow without oxygen. So if you seal this off and let no oxygen get in, you're a lot more likely to be growing bacteria that are dangerous to us, and that is not something we want to do. And then once you've taped it, you put it upside down and you go and put it in an incubator to grow. You put it upside down because there's quite a lot of moisture in here and it's going to be quite warm, and so you'll get a level of a layer of condensation on the lid, and that will form a drop that will then drop onto your agar plate, and it will disturb the nice layer of bacteria you have. So you store it upside down and you leave it for days or maybe up to a week in an incubator with a temperature of about 25 degrees, and it will grow quite nicely at that. The reason why we use 25 degrees, it's warm enough to encourage the bacteria to grow quite quickly, but it is still quite a long way away from body temperature. The bacteria that damage us like to grow at human body temperature, about 37 degrees. So if we go much higher than 25 degrees, again, we risk growing those types of bacteria that are more likely to damage us. Once we've grown our bacteria, what it will look like is, depending on the type of bacteria, but usually the type that we will be using, a white layer, a nice layer that completely covers the top of the agar plate. And we could use that for a number of purposes. We could, not at school, but if you're a scientist, you could take a sample of that and get its DNA out to find out the genetic sequence it has. You could put it under an electron microscope to have a look at the structure of it. You could also use it as a sample to test different antibiotics to see whether the antibiotics that you want to use are going to kill it, whether the disinfectant you want to use is going to kill it. And that's the main purpose that we'll be looking at in next lesson for why you want to grow bacterial lawn. Anything else I wanted to say about this? No, I think that's it. I'm going to put a link to another video as well so that you can see how it's done with the proper equipment as well. I just wanted the chance to be able to talk you through it and make sure that we covered all the bits that are really important for you. Okay, bye.